So good morning, everybody. I'd like to get our webinar started. So again, good morning, good afternoon, and good evening. Welcome to the third and last webinar before the October 11th and 12th workshop on the state of the science of different gastrointestinal model systems, their strengths and limitations, and establishing confidence in existing models. So this webinar features 2 talks on food antigen transport across the intestinal epithelium and induction of allergen-specific antibodies. All right, so my slides are not advancing. Let's move. Sorry about that. We are going to see if I can shift screens. There we go. So if you have participated in some in our prior 2 webinars, you will have seen some of this background information. So very briefly, with the potential of orally administered substances and their metabolites being toxic, absorbed and systematically distributed, methods that can screen for this potential are clearly desired. So with the advent, growth, and use of new approach methods, which we'll call NIEHS, we have seen that these approaches, and for example, like IATA's defined approaches that we've seen with skin sensitization, with oral and ocular irritation, their ability to offer increased human relevance and better prediction without the use of in vivo studies. So as I mentioned, this is the third of a 3-part webinar series that is laying the foundation for the upcoming October 11th and 12th workshop. So I'd like to recognize the organizing committee for both this webinar and this workshop. This was a very active committee, and you can see there was actually very broad interest in this topic. So as I mentioned before, we have this workshop, which is next week. You can participate, you can still register to participate virtually in the workshop. We've actually closed for the in-person workshop registration. So that registration can be found on the NIEHS website, and also on the NIEHS, .nih.gov gut models link. So this is the agenda for today's webinar. And before we begin, I have the first presentation, actually, I have a few housekeeping slides. First, if you're not familiar with this platform, please rename yourself after your name, put down your affiliation. So whichever, what agency or company, or organization, or university you work for. And also most of the controls that you will find are on the lower panel of this platform. So for example, please raise your hand or use the Q&A for putting in a comment. And that's actually what our next slide is. So if there's time, each speaker will address some clarifying questions after their presentations. So again, please raise your hand or use the Q&A for questions. And lastly, this webinar is being recorded. So the recordings and the presentations for the first 2 webinars are available. They are on the website, and the link is provided here. And again, this is the link that you can use to register virtually for the workshop presentation. Excuse me, for the workshop to participate virtually. So let me get on to our speakers. Our first speaker is Dr. Simon Hogan. He is the Asquith Research Professor of Food Allergy and Professor of Pathology at the University of Michigan. He has more than 20 years of experience in basic immunology research, concentrating on delineating interactions at the immune gastrointestinal epithelial interface and how alterations in these interactions can predispose to and modulate food allergic reactions. So thank you very much. And Simon, please take it off.
Okay, sorry. Thank you, Joanna. Can people see my slides there?
Yes, we can. Yep, they're perfect.
Appreciate it. It's thank you for that kind introduction, Joanna, and it's a privilege to be here. And what I'm going to do today is I'm going to talk to you, describe to you some work we've been doing, where we've been investigating what what we initially thought would be a simple process, but worked out to be more complicated. The mechanism by which food antigens actually cross the intestinal epithelium, and what are the consequences of these antigens crossing the epithelium in normal healthy individuals, and also in food allergic individuals. As we have a strong interest in food allergy, we're very intrigued to see whether the processes of antigen movement across the epithelia were similar between allergic and non-allergic individuals. So to get us on page, I first want to show disclosures. The group does have partial grant support from Regeneron Pharmaceuticals. And I want to primarily get us focused on the intestinal epithelium. And really, to get to the epithelium and get to the importance of the epithelium, we have to kind of go back to the 37,000 feet level and look at the small intestine. And the small intestine really is kind of the primary focal point for the degradation of nutrients and food products that are critical for metabolism. And the reason this is that a lot of the nutrients that we actually eat are in too complex of a form, or they're insoluble or undigestible, and they actually have to be degraded. And the gastrointestinal tract is essentially an evolved system that goes through and permits a process of mechanical and enzymatic digestion to simplify the nutrients and permit absorption and utilization for survival. And so while the larger compartment of the GI tract, the mucosa and so forth, I'll just make sure I get this marker. I think that's the marker there. Oops, I'm sorry. I won't worry about the marker. If you get down to the epithelium digestion, and the epithelium itself is really the focal point of absorption. So the priority for the epithelium is actually surface area. And you can see here, if you look at the actual small intestine, you can see that you have the villus, and you can see the villus themselves actually have multiple protrusions, which is which you have your villus crypt, which maximizes surface area. And the actual villus itself is covered by a simple epithelia, a single layer of epithelial cells. What's important, and here's an example here of a histological stain biopsy, a duodenal biopsy from a human patient. And you can see the villus structure here of the simple epithelia, and you can see that there's various epithelial cells that are structurally different. And the open cells here are actually goblet cells. But the epithelia itself consists of multiple different specialized cells that have evolved to have individual roles in maintaining the gut epithelium, the gut interface. Those being Paneth cells that you have present in the epithelia, and the Paneth cells are primarily involved in defense against the microbial antigens that are present in the gut. You have goblet cells, which essentially provide the lubrication. Provide the lubrication to permit
lubrication to commit.
Can people, Parry, can you manage that response, please? The goblet cells provide the mucus. Enteroendocrine cells are also another cell population that are very important in providing hormonal regulation of gut content. And then you also have enterocytes, and the enterocytes are another secretory lineage that are involved in metabolism of proteins and carbohydrates. So you have to remember that the epithelial layer itself actually is semi-permeable, because it plays an important role in acting as a fence to protect us from some luminal contents, such as carbohydrate, protects us from some luminal contents, such as microbial antigens, pathogens, and viruses, where it has to prevent uptake. However, in other situations, such as carbohydrates, drugs, and chemicals and vitamins, it actually has to allow permissible uptake for metabolism. And when this actual barrier breaks down, this is where we seem to lead to disease exacerbation, where we get toxins and innate antigens cross the epithelium and engage with the immune compartment. And this is what's considered to be primary driver of various GI and systemic autoimmune diseases, such as food allergies, inflammatory bowel disease, and metabolic disorders. The content of the gut lumen also plays a really important role in immune education. We can't forget this. And it's over the last 10 to 20 years, I think it's become very paramount that we're learning that a lot of the microbial organisms and the metabolites from these organisms play a very important role in immune education, but also in driving immune tolerance, and particularly, for example, to dietary antigens, where these dietary antigens in the presence of the metabolites develop what we call an oral tolerogenic response, in that we become immunologically unresponsive to these food antigens. While we're very familiar with the immune mechanisms by which this occur, we're not as familiar with the way in which the dietary antigens actually cross the epithelia. More often than not, you'll see in presentations, for example, here, and also in our review articles, where we just generally show an arrow where the antigens move across. So we became very interested in this actual process and wanted to understand more how gastrointestinal dietary antigens cross the intestinal epithelium. So the way we simply addressed this was we used this 2-photon imaging approach, where we had a live mouse that was anesthetized. We surgically removed out the small intestine and put it on a microscope, put it on a 2-photon microscope, and we're able to do imaging actually on the gastrointestinal compartment. And we can do this imaging on any part of the gut. What we do is we micro-inject various food antigens or fluorochromes and also fluorescent agents to give us orientation actually of what's going on in the gut. And if you can see this still image here, cross-section of the smaller intestine, you can see the red compartment, which is dextran rhodamine. That's a fluorochrome demonstrating the lumen of the gut. If you see the blue coloration, that's a nuclear stain. That is showing the actual epithelial cells in the villus. And in the black area, this is actually self, or this is the self side of the villus. So if you imagine this is cutting a transverse section of the villus, so you're looking down in the villus. So what we did was we took fluorochrome labeled antigens, egg, peanut, cow's milk, injected them into the lumen of the gut and simply monitored movement of these antigens. And what you can see here is that we could start to see over time, as we're sectioning here in the 2-photon imaging, these antigens seem to be moving across the epithelium. You can see the absorption and the movement across the epithelium into the self area, which suggested that the antigens are being taken up by the epithelial cells. We first asked the question of whether this is specific for any type of antigen. And so we labeled peanut, egg, and milk clinically relevant food antigens, and showed that no, this process seemed to be indiscriminate about what type of antigen. All antigens seem to take the same process, where they seem to be taken up by the intestinal epithelial cells. We then wanted to ask the question is, well, what are the actual cell populations that are actually contributing to this process? So we did immunostaining, and we primarily looked at did staining for MUC2, which is a goblet cell indicator, and we could show co-localization of the milk, for example, in MUC2 positive cells, which suggested in goblet cells. So this identified to us that in fact these goblet cells had the capacity to rapidly take up food antigens within minutes of exposure in the small intestine. And this was very reminiscent of a study that happened before our investigation by a group, Rodney Newberry and colleagues at WashU, that identified this process of goblet cell antigen passages. And this description of goblet cell antigen passages was there's antigens taking up goblet cells taking up innate antigens by a muscarinic receptor dependent mechanism. And there seemed to be commensal antigens and so forth that could be taken up by these goblet cells, and they were able to be passaged to immune cells, particularly they demonstrated dendritic cells. So we thought this is a similar mechanism where dietary antigens are being taken up by the same process. Over the last probably 5 to 10 years, there's been a lot of interest in these goblet cell antigen passages, and what's been revealed primarily by the Newberry group is that these goblet cells, when they secrete the mucins, have the capacity to, at the same time, then take up the antigens through this fluid phase bulk endocytosis process. that indiscriminately takes up luminal antigens and then shunts the antigens from the apical membrane of the cell down to the basolateral membrane of the cell, but also is able to passage the antigen to various compartments of the cell as well, the lysosome, potentially the Golgi, and the mechanism that we were interested in, the transcytosis of antigen to an underlying immune cells in the basolateral compartment. They've also done studies to actually show that these antigen passages seem to have the capacity to spatially and temporally change their activity in the GI tract. So in the very early on in life, these antigen passages are predominantly present in the small intestine, and then following exposure to EGF in the mother's milk, this turns off these antigen passages in one GI compartment, but as the EGF goes down, these antigen passages turn on in the large intestine, and then through mechanisms regulated by the microbiota, can switch off in the colon and switch back on in the small intestine, showing that there's these mechanisms where we can control spatially and temporally these antigen passages, which we think it relates to the ability of these antigen passages to shunt antigen to the immune compartment and regulate tolerogenic mechanisms, whether it's tolerance to the commensal microorganisms or tolerance to food antigens. So that is one mechanism that we identified, and there's been several mechanisms that have been proposed by which dietary antigens can be taken up by the epithelial cells in the immune compartment, and I'm going to briefly describe them. One is the more well-described process of M cells that are positioned over the follicular aggregates. M cells take up antigen and present them, present the antigens to the immune compartment right under the epithelial cell, the invagination of the M cell, where there's dendritic cells, B cells, and T cells, and that can passage antigen to these APCs that then are thought to be involved in tolerogenic responses. Enterocytes have also been shown to contribute to dietary antigen uptake, but this is primarily through degradation processes or metabolism and simplifying these complex nutrients and then being able to shunt them across the epithelium through various mechanisms. We have the goblet cell antigen passages that I just described through goblet cells. There's also known to be these transepithelial dendrites that actually have the ability to send out a pseudopod between the epithelial cell, maintaining the barrier, and be able to sample antigens on the luminal side. These transepithelial dendrites are thought to primarily occur in the ileum and are quite rare. And then the final kind of mechanism is the mechanism of indiscriminate paracellular leak, where these antigens can passage actually between the epithelial cells. So what are the unique characteristics of the frequency and the spatial and temporal localization of these mechanisms? And you can see that these mechanisms seem to these processes seem to discretely be able to sample antigens of different complexity. The M cells seem to be able to sample soluble antigens or aggregated proteins, and are primarily involved in secretory IgA immune complexes because of the Fc receptor expressed on the M cells. They predominantly or only occur over follicular aggregates and Peyer's patches, and they're about, at the frequency is about 10%. If you look at goblet cell antigen passages, these, which I described, these soluble proteins and peptides of lower than 100 kilodaltons, they're present in the small intestine and distal colon, and they're spatially and temporally regulated. And there's probably about 3 to 5 gaps per villus, showing the frequency of these antigen passages is quite high, and they're distributed throughout the gastrointestinal tract. You have the transepithelial dendrites that I mentioned are in the terminal ileum and are very infrequent, and these primarily involved in sampling bacteria and debris. And then you also have the paracellular mechanism, which involves small peptides that can also occur throughout the GI tract, but there's intrinsic complications with this mechanism given the paracellular leak and innate antigen exposure. So if you consider that a lot of the food allergens that primarily drive food allergy are all in the proximity of around 10 to 50 kilodaltons, we were very interested in the mechanism of goblet cell antigen passages and actually contributing to dietary antigen or food hypersensitivities. And so we primarily focused on this mechanism as a process by which to look at how dietary antigens can alter immune environment and immune activation. So with the first question we asked is, do GAPs contribute to food allergic responses? We took mice that we had spent many years developing that we were able to induce a food allergic reaction in. And what we were able to show is that mice, the epithelial cells, and this is image, the movie's not going to work, but essentially what we're able to show is that in food allergic mice, the frequency of these goblet cell antigen passages is highly upregulated. So this says two things. One, in the epithelial antigen sampling in the food allergic mouse is altered, and the ability to absorb antigen is higher in food allergic mice than naive mice. So goblet cell antigen passages are increased in the small intestine of food allergic mice. We then asked the question is, well, we knew that goblet cells contributed to antigen passages in a homeostatic state. What happens in the food allergic state? And essentially what we were able to see is that not only does the frequency of antigen passages change, but also the landscape and the populations that contribute to the antigen passage change. In that we were able to demonstrate that not only goblet cells contribute, but endoendocrine cells and Paneth cells also are contributing now to the antigen passages. And the spatial regulation changed because we could now see it in the crypt epithelium as well. So this indicated that multiple cell populations, particularly of the secretory lineage, were contributing to these antigen passages in the food allergic condition. And you can see this here, where in the food allergic mice, not only is there higher frequency, there's other populations that are contributing, but in the homeostatic naive state, it's only presence in the villus and they're only goblet cells. So under food allergic conditions, antigen passage patterning and landscape is dysregulated, and there's multiple intestinal epithelial cell populations contributing. So we termed this SAPs, secretory cell antigen passages, because there was all the secretory lineages were contributing to the antigen passages in our food allergic mice. So we come to the conclusion that under steady state, goblet cells were primarily driving antigen passages, and it was goblet cell restricted, and they seem to be contributing to passage in the antigen to dendritic cells. Under the food allergic state, we'd seen that more epithelial cells, secretory cells were contributing, but we weren't sure whether there was a change or alteration in the immune cells that were receiving the antigen. So we knew that under food allergic conditions, that a food allergic reaction happens very rapidly after antigen exposure, within minutes. And we anticipated that if this response is occurring within minutes, it's not likely that the goblet cells or the secretory cells are presenting antigen to just the dendritic cells. We'd anticipate that it must also present antigen to the other immune cells, and we hypothesized it was mast cells. So we asked the question, do food allergens passage across secretory antigen passages and stimulate IgE mast cell responses in vivo? So we took a food allergic mouse and we simply stained for the presence of mast cells. And you can see the frequency of mast cells, which are these CAE or red positive cell stains here, are very high in the small intestine of a food allergic mice. And what you also notice is that these mast cells seem to be predominantly localizing around these secretory epithelial cells in the small intestine. We did immunofluorescence analysis, and we essentially labeled the food antigen, which is dextran. We labeled goblet cells, which are green in MUC2, and we labeled mast cells. And what you can see here is that, particularly in this position here, you can see a goblet cell that's taken up antigen. The antigen is being passaged through the goblet cell and seems to be co-localizing with mast cells by this crimson stain, where you've got red and white positive staining cell co-localizing the antigen with mucosal mast cells. If we take the mast cells out of the gut of these mice and look for the presence of antigen, we can see the presence of antigen on the surface of mast cells. So this clearly shows that if we give antigen within minutes, these goblet cell antigen passages or secretory antigen passages have the capacity to passage antigen directly to mast cells. So SAPs acts as a conduit passaging luminal food antigens to mast cells in the small intestine. We asked the question is, how important is this step or process in induction of a food allergic reaction? And the way we did this is we genetically engineered mice that essentially were not able to activate SAPs. So there was no secretory antigen passages in these mice. And what we predicted was if we block these secretory antigen passages, we should block antigen movement across the epithelium, block mast cell activation, and thus block a food allergic reaction. And indeed, that's what happens. If you look at mice that have SAPs, if you give them antigen within minutes, you see mast cell activation as the surrogate marker of mast cell protease 1. And you also see evidence of hypovolemic shock or systemic shock response where the mice lose temperature within 15 minutes, which we know is mast cell dependent and evidence of an IgE-mediated reaction. If you take away secretory antigen passages, this does not happen, showing that SAPs act as conduits of luminal food antigens and that inhibition of these SAPs prevent IgE-mediated food-induced reactions. So what we were able to show now is that these antigen passages are very important in moving dietary antigens across the epithelium, but they seem to be spatially and temporally regulated and can control oral tolerogenic responses, but also contribute to food allergic responses. Well, next interested in the question is, well, if these antigen passages can passage dietary antigens, do they contribute to food sensitization? And we know that food sensitization is related to the presence of certain immune molecules, such as the cytokine IL-4 and IL-13. And we knew that the mechanism by which SAPs occur is related to IL-13 and the IL-4 receptor alpha. So we predicted that, well, if we get a change in the immune environment in the gut, do we transition from GAPs to SAPs? And do these SAPs now be able to alter oral tolerogenic responses versus food sensitization responses? And why we're very intrigued by this is simply because of the fact that a number of the IL-4 receptor family members, SNPs in these molecules like IL-13, STAT6, and IL-4 have been linked with predisposition to food allergy. So we thought that in this context, if you have a SNP or a mutation that have heightened IL-4 signaling, this could lead to a change in your antigen passages and contribute to sensitization. So to test this question, we took a mouse that has a mutation that's similar to mutations that occur in the IL-4 receptor alpha in humans. And this leads to heightened IL-4 signaling, and simply ask the question that do these mice at steady state have GAPs or SAPs? And do they become sensitized to oral antigens following oral exposure? So we took the mice, studied the antigen passages, and gave them food antigens. So a short answer is what we could show was as we've seen before, in normal wild-type mice actually only have GAPs. And if you give them muscarinic antagonists, you can activate these GAPs. There's no non-GAPs, so no secretory antigen passages. If you take mice that have a mutation in the IL-4 receptor alpha, these mice at steady state not only have goblet cells acting as antigen passages, they also have other non-goblet cells acting as antigen passages, and the frequency of these cells contributing this response are increased. So this suggested to us that if you simply mutate the IL-4 receptor alpha in mice, these mice have an altered landscape of antigen passages in the small intestine at steady state. If we then expose these mice to egg, peanut, or milk, at least to egg and peanut, we can show that oral exposure and activating of these antigen passages can lead to the presence of immunoglobulin, IgE, and IgG1 in these mice. And if you then, after they develop immunoglobulin, you orally expose the mice, you can now demonstrate evidence that these mice will have a food allergic reaction. They will start to develop hypovolemic shock following antigen challenge within about 15-30 minutes of antigen challenge. And they'll start to demonstrate evidence of diarrhea response as well, showing GI symptomology in these mice. And you can also show evidence of mast cell activation. So these studies led us to conclude that if you have alteration in the IL-4 receptor alpha or SNPs in molecules that have heightened Th2 immunity, this alters your antigen passage formation in the gut at steady state and can predispose to food antigen sensitization. Obviously the question is, what about in humans? So we had been collaborating at this time with some colleagues at Cincinnati Children's, an outstanding pediatric gastroenterologist, surgical enterologist, Michael Helmrath and Max Mahi, who was a postdoc in Michael's lab. And they developed this very elegant system where you could take pluripotent stem cells, human pluripotent stem cells, develop human intestinal organoids, and you transplant these human intestinal organoids into an NGS immune deficient mouse into the kidney capsule and be able to watch these human intestinal organoids grow. And here's an example here where you actually transplanted the mesentery actually of these mice, NGS mice, and you can see that these human intestinal organoids will grow. And when they grow to a certain size, we can then start to do the 2-photon imaging to actually look at the ability of these epithelial cells to present antigen. And so, as I described, we take the HIOs and leave them in the mouse for about 10 weeks, and these HIOs start to form normal epithelium, human epithelium. Here you can see the goblet cells and the villus crypt. And then if you actually do 2-photon imaging of the human intestine transplanted into the mouse, you can see that, yes, they do form antigen passages. And here's an example here where we have milk and dextran co-localizing within goblet cells. And if we activate the IL-4 receptor alpha, we can see enhancement not only of goblet cell antigen passages, but also of other secretory lineages that are contributing to this response. So in conclusion, this study suggested to us that food, dietary antigens and dietary allergens can cross the epithelium by multiple immune processes. In particular, in the small intestine, we've identified that these antigen passages are very important in acting as conduits for antigens to move across the epithelium and present to the immune compartment. We can show that the antigen passages are spatially and temporally regulated, and the patterning and the landscape of these structures are altered under food allergic conditions and temporarily regulated in the small gut, small bowel, and the large bowel. We're able to show that these secretory antigen passages at least can contribute to IgE mast cell mediated reactions. And if you inhibit these responses, they can inhibit food allergic reactions. And we also show that human intestinal tissue has the capacity to also form goblet cell antigen passages and secretory antigen passages. So these are the acknowledgements, collaborators in WashU with Rodney Newberry and his team, outstanding team, and also my outstanding team here at Michigan, where we continue to do this work. And if I have time, I'd be happy to take any questions. Thank you.
Thank you, Simon. Actually, because of the time, we'll probably move on to our second speaker, but I would encourage participants to post questions in the Q&A, and Dr. Hogan can address those while we're moving through our second presentation. So I'd like to introduce our second speaker. Dr. Sarita Patil is an assistant professor of medicine at Harvard Medical School and an assistant physician at Massachusetts General Hospital, where she is a member of the divisions of allergy and immunology in the medicine and pediatrics departments. She is co-director of the adult multidisciplinary eosinophilic esophagitis clinic at Mass General, which she helped develop for treatment for eosinophilic gastrointestinal diseases. Her laboratory in the Center for Immunology and Inflammatory Diseases and the MGH, so Mass General Hospital, Food Allergy Center, focuses on understanding antibody and B cell responses in both the initiation and treatment of allergic diseases, with a particular focus on food allergies. Most recently, she has been investigating the mechanisms of antibody-mediated protection in oral peanut immunotherapy, as well as focusing on the development of antibodies to allergens. And thank you very much, Dr. Patil.
Sorry, thank you so much for that really kind introduction. I'm very excited to be here. So I'm going to carry forward the story that Simon started to tell in the field of food allergy.
Your PowerPoint is in like presenter's mode, so you need to swap it.
Let's see here. Does that work better?
No, not yet. Try to swap display.
I just swapped it. It must be a sharing issue.
Yes, we're not seeing the full screen.
Let's see if I can do it this way. Is that right now?
Now it's working. Yeah.
Thank you. Sorry about that technical glitch. So I just wanted to kind of, put a little bit of a clinical spin here on, why we think so intensely about allergenicity in the context of food allergy, and, raise the point that the incidence of food allergy has risen just, exponentially over the last decade, far beyond what we could have imagined. And, the food allergy touches all of our lives in multiple ways every day, not just, in the lives of our patients who are at risk for, reactions recurrently due to minute exposures to things that are commonly found in their environment, but also, the entire, population is affected, particularly our youngest members, our children, in whom food allergy continues to increase. And so, when we ask the question, what is allergenicity and how does it get into the body and why does it manifest in these maladaptive responses? I think that these questions are fundamentally, critical to our ability to understand and innovate in this area. And so in the context of food allergy, to kind of reorient this discussion a little bit, when patients with IgE-mediated hypersensitivity are exposed to minute exposures, it can trigger cross-linking of allergen-specific IgE on the surface of mast cells, causing those mast cells to degranulate, release their mediators, and induce a multi-systemic response, including potential for anaphylaxis, which can be fatal and involves multiple systems at the same time. And for the longest time, our only remedy was to suggest avoidance, and in the case of an accidental exposure, the use of epinephrine, which can prevent that anaphylactic shock, or we can stop the anaphylactic shock. And so how does this entire process get initiated? So we just heard from Simon how food allergens can travel through the epithelium to activate the adaptive immune system. And I'm going to take that to the next step and really dissect the mechanisms by which this happens. We think that in patients who have a predisposition to seeing these allergens, they have pathogenic T cells that will then present these allergens in addition to pro-Th2 cytokines like IL-4 and IL-13 that can then drive the switching of memory IgG1 cells that are Th2 primed, meaning they have upregulation of the IL-4 receptor, for example. And then these in the lymph node will receive help from T follicular 13 cells, which then drive IgE class switching and the production of allergen-specific IgE. And this process of developing allergen-specific IgE can then set up those individuals for having a systemic anaphylactic reaction. It turns out, though, that we can modulate these Th2 subsets using peanut oral immunotherapy. And in fact, if we treat those patients who have allergen-specific IgE and react to peanut with incrementally increasing doses of the allergen, peanut in this case, orally, with careful observation, of course, we can actually change or delete or energize this particular subset. And instead of driving IgE, we can drive IgG. This idea or this paradigm that in patients with allergen-specific IgE, we can actually deliver the allergen through immunotherapy to induce allergen-specific IgG has been one that's been studied and established in the field since the 1950s, when Robert Koch first discovered this phenomenon. But in the context of food allergy, we realized that the sheer measurement of allergen-specific IgG didn't seem to completely explain the phenomena of tolerance. And what I mean is that in food allergy, we can granularly measure how much clinical efficacy oral immunotherapy has by serial challenges. So these patients, we can find that their exquisite sensitivity to their allergen is modified over the course of immunotherapy because their post-immunotherapy challenge, they can tolerate a higher level of allergen than before. However, we found that if you stop immunotherapy, some patients can continue to ingest the peanut at a fairly high level and not have a reaction. So we call those patients having sustained unresponsiveness, although, and another set of subset of patients, a much larger subset of patients actually lose their hard-won tolerance, right? And we call those patients who had only transient desensitization, shown in red in these two examples of clinical trials where they examined these two clinical outcomes and found that the levels of allergen-specific IgG4, so peanut-specific IgG4, was the same in both groups. So it didn't seem like the allergen-specific IgG4 was really correlating with clinical outcomes. In our clinical trial, we also tried to focus down in on the immunodominant peanut allergen ARA-H2, and again found the same phenomena, that both the patients with sustained and transient responses had the same levels of allergen-specific IgG4 induced. And so we wondered whether it was not a property of the quantity of antibodies, which is what we were measuring, but rather a qualitative property. And what do I mean by that? I mean, was it something about the affinity, the clonality, the diversity, the epitope recognition, or the Fc receptor binding of these antibodies that are induced during immunotherapy, specifically in those patients who have more sustained responses, that might correlate to clinical efficacy or that might drive clinical efficacy? And so to dissect that question, right, we kind of constructed a model in our minds of how allergens and allergen-specific antibodies might interact. And that, at that gut luminal surface that we were examining, those patients with sustained responses, while they would still see allergen, would have these antibodies that would effectively sequester them away from cross-linking IgG on the surface of allergen effector cells, whereas those patients that only had transient responses would still induce an allergen-specific IgG response, but just one that wouldn't be so effective at sequestering allergen. And so to examine that, we really need to dissect how allergens and antibodies actually interact. And so we turned to two clinical trials of peanut oral immunotherapy, both a pediatric and an adult trial, where we were able where patients were able to undergo a high dose course of oral immunotherapy, meaning that they got to a total of 4 grams of peanut allergen during the course of their immunotherapy. They then stopped it for 1 to 3 months, and then we examined whether it had sustained or transient effects. And so we could take a subset of patients who had sustained responses and a subset that had transient responses, so that we could finally start to dissect their allergen-specific antibodies using a technique that we pioneered in my lab in 2015 to use allergen-specific multimers, where we took ARA-H2 and complexed it with a fluorescent molecule to be able to detect allergen-specific B cells with an extremely high specificity, about a 90% specificity, which let us look at the kinetics of allergen-specific B cells during immunotherapy. So in these patients who are receiving oral immunotherapy and who already have IgE, we could see that as we progress through increasing doses of immunotherapy early, there was an early and transient allergen-specific memory B cell response that emerged and then slowly waned over the course of immunotherapy. So we took allergen-specific B cells at this peak time point, sorted them into individual cells to recombinantly clone their heavy and light chains. We did that with thousands of sequences and ended up with about 79 monoclonal antibodies to characterize. And, I should point out that while, and these monoclonal antibodies were directed to ARA-H2, and while we knew that ARA-H2 was an immunodominant allergen, it had been proven to be very difficult to characterize. So we only understood the sequential epitopes of ARA-H2. Getting an X-ray crystal structure of ARA-H2 was very, very difficult. In fact, the only X-ray crystal structure we had to date when we started this project was one that Jeffrey Mueller at NIEHS had produced in combination with a large carrier protein. So the first thing we did is use sequential peptide epitope mapping using peptide microarrays. This is a technique that's been pioneered many, many years ago, where we take overlapping peptide sequences of ARA-H2, put them on a glass slide to see which monoclonal antibodies bind. And this heat map shows the intensity of the secondary antibodies used to detect those monoclonal antibody binding. And we found 4 main sequential epitopes. So the first one, and the most abundant one that we found, was this epitope called DPY hydroxylproline S epitope. It's a repeating motif that's found on a loop region of ARA-H2 that's extremely flexible. And it's actually unique to ARA-H2. There are several other 2S albumin seed storage proteins in peanut, but only ARA-H2 has this loop region. And what's really interesting is that because there are multiple motifs within the peanut, within this ARA-H2 immunodominant peanut protein, you can actually use a multiple motif segment, just that loop region of ARA-H2, and actually activate allergen specific activation of effector cells by cross-linking that IgE. The three other sequential epitopes that we identified are also ones that have been previously identified by peptide microarray of polyclonal serum from peanut allergic patients. So we felt very confident that these sequential epitopes were highly relevant in the field of peanut allergy. And we also knew that there are newer techniques for sequential epitope mapping of polyclonal IgE that have shown that detection of these peptide of antibodies that bind to these peptides can actually increase diagnostic specificity for peanut allergy. So antibodies that recognize these sequential peptides are found in multiple patients in peanut allergy. But to understand the conformational epitopes, right, those antibodies that did not bind to any peptides on this microarray, we turned to biolayer interferometry and epitope binning, where we used a sensitive sensor, so a sensor that was a streptavidin sensor that was attached to biotinylated ARH2. We dipped the sensor into primary antibody where we saturated that primary antibody to detect whether a secondary antibody could bind, in which case it would be binding to a separate epitope of ARH2 or the same epitope, in which case it wouldn't cause a deflection in the wavelength of light. And so we looked at the response rates and could group them into three main groups, identifying three main conformational regions of ARH2 that we very creatively named bins 1, 2, and 3, shown in blue, orange, and red on this map. So looking at all of the recognition by multiple monoclonal antibodies over these two groups of patients, either those patients with transient responses on the top or sustained responses on the bottom, we could look at the epitope specificity across these patients. And at first glance, it's obvious that both patients with transient and sustained responses have antibodies that recognize multiple epitopes. So sequential epitopes are recognized in both groups, and conformational epitopes are also recognized. The most commonly recognized epitope was bin 1, then bin 2, and bin 3. What was interesting about bin 3 was that antibodies that recognized bin 3 were only found in patients who had sustained responses to peanut oral immunotherapy, and that was our first clue that there might be something very interesting about the way these antibodies recognize their epitope. We also found that these epitopes were distinct and discrete over ARH2. So when we took this sensor that was attached to biotinylated ARH2, we could dip it serially into wells that contained antibodies that can bind to each separate bin, and found that indeed we could load 4 different monoclonal antibodies at the same time. So we can see deflections when we can bind bin 1, 2, 3, and then separately that loop region of ARH2. These antibodies that we characterized were extremely high affinity ARH2. So they had a sub-nanomolar affinity to ARH2 that we calculated by biolayer interferometry, shown here. It was very similar across all 3 bins. And there was no difference in the affinity in these high affinity antibodies, whether they came from patients who had sustained or only transient responses. So we finally did manage to identify the epitope-paratope interactions between these monoclonal antibodies and ARH2 using X-ray crystallography. So our colleagues here, Jeffrey Mueller and Lars Peterson, along with their talented postdoc, Chun-Ki Min, and in collaboration with Anna Pomas for InBio, we were able to find 2 FABs of 2 monoclonal antibodies. So the FAB of the antibody that recognizes bin 1 is shown in blue, bin 3 is shown in yellow, and their epitopes on ARH2, shown in the middle here in white, are colored in red and blue respectively. And when you look at all of these, we can identify the epitope for bin 1, 2, 1, 3, 2 would be tucked underneath, and the loop region, which doesn't show up in X-ray crystallography as it's so disordered, is on the top there. And so this allowed us to identify not only where these epitopes were localized, but also the fact that these epitopes were recognized by both the heavy and the light chains of these individual monoclonal antibodies. And the way that we were able to detect them was because these antibodies recognize multiple loops within ARH2 as well. So they're very complex and very high affinity due to the way that the epitope and paratope interact. Ultimately, through a number of other experiments, we were actually able to fractionate these epitopes into 4 main conformational epitopes of ARA-H2. Epitope 1.1 and 1.2 were overlapping epitopes that we identified in that bin1 region. And then there was epitope 2 and 3. And we only found antibodies that could recognize epitope 1.2 and 3 in patients who had sustained responses, but not in patients who had only transient responses. And so we wondered whether it had to do with the way that they recognized the allergen. And so we looked at the ability of these antibodies, these monoclonal antibodies, to functionally inhibit IgE binding. So we use pooled IgE here in order to increase the diversity of IgE recognition. And first, we use monoclonal antibodies to detect or to prevent the binding of serum IgE, and looked at the percent inhibition. Those antibodies that recognize conformational epitopes can inhibit more polyclonal IgE recognition of ARA-H2 than those antibodies that recognize sequential epitopes. And breaking it down by each monoclonal antibody within each region, we could actually look at mechanistically why this might be the case. So here are the antibodies that recognize that bin3 epitope. So this antibody recognized the entire bin3 epitope shaded in red here. And there are 2 sequential epitopes boxed in here that also recognize an overlapping region. And you can see that this monoclonal antibody can block significantly more polyclonal IgE than these 2 antibodies that recognize the overlapping sequential epitopes. We found the exact same phenomena again in epitopes 1.1 and 1.2. It's interesting to look at this antibody that recognizes epitope 1.2 because it inhibits a huge amount of serum IgE binding in comparison to the overlapping sequential epitope that is localized in a similar region. So taking this all together, we put this entire story together as there are these unique antibodies in patients who have sustained clinical efficacy that can block multiple IgE epitopes. So not only those antibodies that recognize similar conformational regions, but also epitopes that recognize sequential regions that might be overlapping in the same area, which allows them to continue to block IgE-mediated activation of effector cells even after immunotherapy has ceased, and we see those total IgG4 levels start to wane. And so this confers long-term clinical efficacy after immunotherapy. Now, one of the big debates in the way that antibodies recognize allergen is the fact that we typically think of antibody repertoires as highly individualized. The whole process of our immune system recognizing allergens, those complementary determining regions, which bind from the antibody to the allergen, right? And that mutate over the course of affinity maturation, somatic hypermutation, that those, the introduction of those mutations is highly individual. And so we would expect that every individual's repertoire would be highly unique to their own body. Interestingly, in 2015, when we started looking at ARA-H2 specific B cell sequences, we started to notice a very interesting pattern. That is that we started to see similar stereotyped sequences across multiple patients. So those BCR sequences that shared the same rearrangement of their variable and joining regions and had a highly homologous CDR3, so the main region that usually recognizes antigens, a very highly homologous CDR3 region. And if you even looked at the other CDR regions, CDR1 and 2, they also were very, very similar in their amino acid properties, suggesting that there was convergent evolution of these antibodies towards this small 18 kilodalton immunodominant protein, peanut protein ARA-H2. Subsequently, multiple other people have found these same sequences within the repertoires of peanut allergic patients, whether we look at single cell sequencing of IgE positive B cells, that was work that was done by Derek Rue, or whether we look at the gut of peanut allergic patients and find an IgE producing B cells, highly similar B cell sequences again to the sequences that were previously identified in a completely separate cohort of peanut allergic patients. And this work was done by Scott Boyd at Stanford. And so in our most recent paper, we validated that these convergent antibodies are not just convergent by sequence, but are also convergent in the way that they recognize the allergen. So all of those antibodies are actually epitope specific as well. So this idea leads to the conclusion that on a population basis, many people are recognizing this immunodominant peanut allergen with their antibody repertoires in a highly similar, stereotyped, convergent fashion. We were also very interested in trying to understand whether this mechanism of neutralization is true within a larger population of peanut allergic patients that are treated with oral immunotherapy. And so to do this, we leveraged that inhibitory, that IgE inhibition ELISA that we had created previously, and used our two neutralizing antibodies highlighted here in green versus the non-neutralizing antibodies. So these are conformational epitopes. These conformational epitopes of 1.1 and 3, sorry, 1.2 and 3 here on neutralizing antibodies, and then 1.1 and 2 on the non-neutralizing antibodies. So combination of antibodies that recognize these are shown in green, and those other ones are shown in yellow, and you can see that they inhibit IgE more potently than non-neutralizing antibodies. And if we look over time in how well they inhibit IgG4, we find that there is an increase in patients who had sustained responses, suggesting that there's an induction of IgG4 neutralizing antibodies in the serum of patients with sustained responses, but not in the serum of patients with only transient responses. Again, helping us understand how, on a population basis, neutralizing antibodies may be responsible for longevity of clinical efficacy after immunotherapy. And in thinking about how to model this using an effective in vivo model, we turned to passive cutaneous anaphylaxis in a humanized Fc-epsilon-R1 mouse. So because these mice had Fc-epsilon-R1 on their mast cells that was human, we were able to sensitize those ears with pooled IgE from peanut allergic patients, which were challenge proven to be allergic. And using that technique, we could then challenge those animals with either ARA-H2 that was complex with a non-ARA-H2 specific antibody, or ARA-H2 that was complex with either neutralizing antibodies, so again, those antibodies that recognize the epitope 1.2 and 3, or non-neutralizing antibodies that are antibodies that recognize 1.1 and 2. And when we did that, we could then measure the amount of response by measuring the extravasation of Evans blue dye. We found that you see a nice robust extravasation to ARA-H2 with a control antibody, which decreases with specific antibodies, but is most potent with a combination of neutralizing antibodies compared to non-neutralizing antibodies. And then finally, all of this work, identification of the molecular determinants of how those epitopes and paratopes interact, understanding how they might interact on a population basis and connecting to clinical efficacy allows us to, again, alter how the allergen might be interacting with antibodies to test whether whether there are critical residues or critical regions on this allergen, which might drive allergenicity. So in collaboration with our NIEHS collaborators, Jeffrey Mueller and Lars Peterson, Jung Ki-min created a hexamutant, which had mutations within conformational epitopes that we had previously identified that bind to ARA-H2. And when we used the hexamutant compared to ARA-H2 and measured its ability to bind to serum IgE, we found the hexamutant has impaired binding to serum IgE from patients with peanut allergy. And this is across many patients who, again, have challenge-proven peanut allergy. And when we again turn to a murine model of passive cutaneous anaphylaxis, the hexamutant induced a much significantly lower response than ARA-H2 alone, suggesting that these regions, by mutating specific amino acids within these regions, we can actually decrease the allergenicity of ARA-H2. And so just to kind of summarize, we've identified neutralizing IgG4 antibodies in oral immunotherapy, and that these are epitope-specific, allergen-specific antibodies that can block multiple IgE epitopes, and therefore really in a very potent and complete way sequester allergen away from IgE activation of effector cells. And we can show this using in vitro basophil activation, which I didn't have time to actually go into today, but which we use in the first paper. And then also using murine models with humanized Fc epsilon R1 receptor on their effector cells. We also can use other tools like hypoallergens and BCR repertoire analysis to look at convergent evolution and understand allergenicity on a population basis. And so, I think breaking down this concept of allergenicity into sort of the molecular determinants of the connection between the structure and the function of allergens and antibodies and how they interact gives us really critical insights into multiple aspects of how allergenicity might operate in patients, right? And the localization of epitopes not only can identify how epitopes overlap and how relevant that epitope overlap might be for clinical efficacy, but also gives us insights into cross-reactivity and predicting future allergenicity. Understanding the paratopes of antibodies helps us understand convergent evolution and understand how allergens are recognized in patients. And then finally, connecting these features of the epitope-paratope with their functionality, their ability to neutralize and to activate allergen effector cells is really critical to connecting all the different aspects of what drives allergenicity in humans. And so I just wanted to acknowledge that this work has really been a tremendous effort by many, many people in our group. It was spearheaded by 3 postdocs, Dr. Nicola Hood, Dr. Tharun Kiswani, and Dr. Junki Min in our collaborators labs at NIEHS. And this work would not have been possible without our clinical collaborators at MGH and many, many people across the world. So with that, I thank you, and I would be happy to take any questions if you have time.
Thank you, Dr. Patil. Actually, we are over our time for the webinar, but that's okay. I'd like to thank both you and Dr. Hogan for some really very elegant talks, some wonderful data that's here. Certainly, I think providing a lot of information on pathways that result in allergenicity through the GI tract. So I actually have questions, but I'm hoping that you can join our workshop as well as our participants, since I think this will really inform this section of the workshop. So again, I hope everybody can join us for the workshop, which begins on October 11th. And I want to thank everybody, and I hope you enjoy the rest of your day. So take care.